This week, we're all here to talk about invasions. And I have a confession to make. I'd like to start my talk by acknowledging that I'm part of an invasion of Australia. So I live in Darug country and work on the lands of the Bedegal people, and these lands have not been ceded by the traditional owners. So it's my hope that in future, in Australia, we do a better job of repairing the damage that we've caused to First Nations people, and also as biologists that we recognize and incorporate the deep knowledge that they bring of the natural environment. I'd also like to acknowledge the traditional owners of the land on which we meet today, the Kingatanga and Waikato Tanui, and express my gratitude that we can meet in this beautiful place. So I guess what I really want to talk to you about today is 3 ideas in invasion genomics that kind of keep me up at night. So these are things that my group works on and that I think about a lot. And the way the talk's going to go today is I'm going to introduce the characters. So these are the model systems that we use and the people that have been doing the work that I'll talk about today. I want to talk about how we're trying to address some of these ideas. And then I'm going to give at the end a little bit of a plot twist about something recent that we've found that has set us on our back heel and that we've been thinking about quite a bit in the last few months. Okay, so the first system that I'm going to talk, well, one of the systems I'm going to talk about today is the European Starling. And this is a system that I did my PhD on and that my group has worked on intermittently over the years. And most recently, Kat Stewart, who's here with us today, did her PhD on the system. And you can see from this map that the native ranges is shown in blue and the invasive range is in red. And so they've really gotten around the planet very well. And if you look at the dates of introduction that I've just put up on the map, you can see that they span a large time span, but we see these really nice systems where we have in the US and Australia, for example, we have introductions at the same time point roughly and from the same place. So we have these replicated experiments that we can use for evolutionary studies. And we also have these incredibly varied environments. So you'll see there's Fiji introduction in the early 20th century. So this is very different from the native range of the Starling. But also, if you look down here on the bottom of the map, that's not a mistake. That's actually Macquarie Island, also introduced in the early 20th century. So these really radically different environments that are there that we can compare. We haven't had the chance to get samples from those 2 places, and I do hope that's a thing we do in future. So the people that I've worked with in the work that I'm going to talk about today, I just want to introduce because I don't want them to get lost at the end. So that's Bill Sherwin that you see on the top left. He was my PhD supervisor and he's continued to be involved in this work throughout. And next to him is Rich Edwards, who more recently joined our team, and he's worked with us on the genome and co-supervised Kat. And below that are 3 students that have worked exhaustively on the system. So the first is Adam Cardellini from Deakin University. Kat Stewart's in the middle, and Natalie Hoffmeister is there on your right. And she's a PhD student who's recently finished at Cornell that worked on some of their genome resequencing data that we have produced. And we have a genome consortium, which was a very large group of people that it was remarkably nice to see a community come together to make a resource that everyone just wanted. When we first got together, there was no PhD student at stake. We just all really wanted to have a genomic resource. Everyone worked really well together and contributed, and we were able to produce a genome which Kat subsequently improved significantly. The other system that we work on a lot is, and suggested is the cane toad. So if you are from Australia, you probably know a lot about cane toads, but I know there are people here from other places. So the introduction of cane toads to Australia has been one of the most remarkable invasions that has been well studied. But they come from Central and South America, as you can see on this map, in the areas in green. And cane toads, like starlings, are a pest of major concern. They're on that top 100 list that we talked about yesterday. I hope our 2 genomes got captured in that analysis. And they have a well-documented invasion history similar to Starlings. And this is like a massive asset when you're trying to understand evolutionary history. So cane toads left French Guiana, we believe, went to the Caribbean. They were introduced as control, biological control of cane beetles. That didn't work very well. But before they figured that out, they kind of spread them all over the planet. And they went from the Caribbean to Hawaii. And then we heard the other night that 102 came to Australia. And that's true, 102 were brought to Australia, but 1 died. So we ended up with 101. And those were bred up. And their offspring were distributed down the east coast of Queensland. So many, 30,000 eggs in one clutch. So many cane toads introduced to Queensland. And currently in Western Australia, in the Kimberley, we have this range expansion front that is moving rapidly. You can see the years on these lines on the map here, where the range moves every year. And so that range edge has gotten a lot of attention from many people because it's a nice system to look at evolution in novel environments. So there's ample evidence of evolution which has occurred across Australia, which makes this a nice system to ask questions, and a really well-known ecology. And that ecology was primarily produced by Rick Shine and his group. So Rick has collaborated with our group for many years now, and he is definitely my primary collaborator on the cane toad work that I'll talk about. That second guy is Mark Richardson, and he is the one that worked on North Pacific Sea Star and won me some chocolate the other night. Thanks, Mark. He was my postdoc, and he ended up being one of our collaborators on the Genome Project and some of the other work I'll talk about. Once again, Rich Edwards is there. He actually led the genome paper, the initial one, and Carlos Rodriguez Lopez is at University of Kentucky, and he worked with us on microbiome in cane toads as well as some of the epigenetic work. And these are all people that worked in Rick Shine's lab. These guys are ridiculously knowledgeable and have been very helpful with us over the years, helping us preventing us from making mistakes that they all learned years before us, so that when we did things like raise cane toads and run common garden experiments, they sort of prevented us from running afoul. And then these are people that were in my group that have worked on the work that I'll talk about today. So that's Boris Yagund on the end. He's a postdoc in my lab currently, working on some RNA-seq work and DNA methylation work. And then Jack Reed is an honors student that did work on DNA methylation. Dan Selechnik worked on immune function in toads and some RNA-seq work there. This is Gia Zhou, and she worked on microbiome. Rashmi Sarma worked on a crazy common garden experiment that I'm about to tell you about today. Rashmi's just finished her PhD, and if you have a crazy common garden experiment that you need someone to run, she's your woman. Look her up. And Kelton Chung is a current PhD student that's working on improvements to the cane toad genome. And once again, we have a genome consortium. This one was much smaller and much more rapid. We produced a pile of data in a very short period of time and got a genome together so that we could do some of the work that we'll talk about. So what do I think about? I think about changes in diversity a lot. This is something that all of us that work on invasions have thought about these ideas. And we have this basic idea that genetic diversity is reduced in introduced populations. And this came from Allendorf and Lundquist's paper, which was published almost 20 years ago. It's been cited over 1,000 times. And in this paper, as I'm sure you guys have all read and know, they talked about how these introduced populations should have similar problems to small conserved populations. And those are problems to do with population growth and persistence and a limited ability to evolve. But they ask, if that's the case, then why are invasive species that have experienced these genetic bottlenecks, why are they so successful? And that brought this whole cascade of papers talking about genetic diversity. Is it true that we have reduced genetic diversity in introductions? What does it do? Is the genetic paradox a real thing or not? And this really nice paper came out that Katrina Lugash looked at decrease in genetic diversity at introduction and showed that over a wide range of studies that there is a decrease in genetic diversity at introduction. And we got a little bit curious after this paper and thought, well, you know, what if we only look at those species that have shown evidence of evolution in their introduced environment? Do we see a difference then? And we don't. We got almost the same answer. So these two groups of studies that we looked at, there is some overlap between them, but they're not. There are a number of studies in our paper that were not in the original one. So it was interesting that we didn't see that difference, right? What we did see is this huge range of values, which says to me that there's a lot more going on than genetic diversity here. We have these populations with huge amounts of genetic diversity that have come from multiple introductions from different places. And we have these ones where we have essentially no genetic diversity. So these are clonal populations. Now, these are mostly neutral markers, right? That's what we had in those days to work with. We had microsatellites and things like that. And so, you know, how do populations with low or no genetic diversity, how do they adapt? Well, they probably have some diversity, right? There's probably some standing genetic diversity at adaptive loci that were not like assayed by those microsatellite studies. And so my student, Dan Selechnik, looked at cane toads, and he's looked at these different populations along the invasion trajectory. And he looked at heterozygosity, and he showed that, in fact, we do see this reduction along the invasion trajectory, which is what we would have expected. But then when he looked at loci that had evidence of selection, he found something a little bit more interesting. And that is that in the native range, we found the lowest genetic diversity. And when we came to the early introduction sites, we have the highest, and then it trails off as we go across Australia. And that was interesting to us. We thought, well, okay, this is what we've been thinking about, right? There's some adaptive variation here that's really important in the introduction. So, you know, yes, there's probably reductions in genetic diversity, maybe not at those loci that are really important. So then we started thinking about, well, what about epigenetic diversity? Could that compensate for low genetic diversity? So I'm sure you guys have been reading papers about this. We talked about it in 2015 in the paper that we wrote after the Asilomar meeting that, you know, it's quite possible that this could happen, but this is not our idea. Others have talked about it. And since Stu wrote this really nice paper talking about, you know, are genetic bottlenecks real? Is that really a thing? And one of the things they said in that paper is that, you know, perhaps epigenetic diversity can compensate. I think the first paper that actually showed data to suggest this might be true was Andrea Liebel's work on introduced house sparrows in Africa, where she showed populations that had the lowest genetic diversity had the highest epigenetic diversity. And they put forward this idea that maybe that's a compensatory mechanism in those places where you have very little genetic diversity. And since then, this very nice review paper has come out, which has detailed a number of other studies which have found that similar kind of result, that on range edges, for example, where you see low genetic diversity, you see more epigenetic diversity. So what about in cane toads? What do we see? So this is a study that GSO did where she looked at genetic diversity and epigenetic diversity in range edge and range core populations. And what you can see here is that the range edge populations are circled in red, and they have less genetic diversity than the range core population. So that's Queensland in the blue. Not a surprise given what I showed you earlier. So this is a different data set. I showed you earlier some RNA-seq SNPs that we pulled out, and this is MSGBS data. When she looked at epigenetic diversity, she saw no difference between the range edge and the range core. So we're not seeing that happening in cane toads. However, she also asked this question about gut microbiome diversity. And you might think this is a bit nuts, like how do we get there from here, right? Well, it's not so crazy because we know that both genetic diversity and gut diversity are linked to fitness. And we also know that there's this potential for gut microbiome to affect the epigenome of the host, and vice versa, that the epigenome of the host can affect gene expression that can affect the community, the gut community. So when she looked at this, she found, not surprisingly, that the genetic diversity has this massive impact on epigenetic diversity. No surprise there. We all know that that's like a major driver of epigenetic state. But she also found that epigenetic diversity had an impact on the gut diversity of the cane toads, and that was interesting. She also found that there was an interaction between genetic and epigenetic diversity with respect to gut community. So we start thinking now back about the concept that Andrea Liebel put forward about this compensatory idea. So if we drill down into that a little bit, what we see is, so this is epigenetic effect on gut microbiome. That's what you're looking at in this graph. And those different colors represent different levels of genetic diversity. So low genetic diversity is the red line, and you can see that there's this really quite strong relationship between methylation diversity and gut microbiome diversity. But when genetic diversity increases, so when you go down on this axis, so that blue line, for example, is high genetic diversity, you see this, this relationship between gut microbiome diversity and host methylation diversity starts to dissolve. So, does that strong relationship that the genetic diversity has on epigenome, is that what's masking the relationship to the gut community? We really need to look further down the road with this data. We need to investigate different tissue types, because what we've used there is blood. Maybe that's not the right tissue to use, maybe it is, but we need to look further. And also look in populations with higher genetic diversity to see if these effects look similar. And once again, we use MSGBS for this, so probably a better methodology like RRBS might be a little bit, give us a little bit more information. Okay, so the second thing that we think about is DNA methylation and whether or not it might be involved in evolution during invasion. So we asked this question about whether DNA methylation can mediate phenotypic responses to environmental conditions. And we use cane toads to do this. Well, I don't know if you guys know cane toads very well, but they're cannibals. And they're really a pain in the butt to work on for this reason, because they eat each other. And if you have them in big cages with lots of cane toads, you come back one day when you weren't looking and there's one and he's really fat looking at you like, but it's a really interesting characteristic that they have, and I'll explain why. So they're cannibals at all life stages. They eat, the tadpoles will eat eggs. And if you have a look at this picture of these two tadpoles, they are full siblings from the same brood. And T1 grew up in a tank without any older conspecifics. T2 grew up in a tank with older conspecifics. And so what happens is those older conspecifics emit chemical cues that affect the growth and development of their younger conspecifics. And if you look at this effect across the range, you find this really interesting effect. This is our collaborator, Dana DeBoer, produced this paper, where in the native range in French Guiana, this is much less of a thing than it is in Australia. So this trait has evolved since it's come to Australia. And this graph shows that the duration of the larval, that window of time where they are susceptible to that chemical cue, is shortened in Australia. So this is a response that those younger conspecifics have to being in the tank with their cannibalistic older conspecifics. And once again, growth and development are affected, and it's much more so in the introduced range than it is in the native range. So we started thinking about this, and we thought, well, what's happening when that chemical gets emitted? Is it changing the methylation of those younger tadpoles? Is that involved in their response to this threat? And Jack Reed, my student, took populations from either end of the range, and he did an experiment where he exposed them or didn't full sibs to older conspecifics. He looked at tadpole growth, and he looked at the DNA methylation of those tadpoles. And this graph shows that, in fact, it does affect phenotype. So if you have a look here on the range edge, you can see that if you've been in the tank with an older conspecific, it's affected your mass. We didn't see the effect in this experiment on the range core. And we do think that this is a continuum, so that this cannibalistic response gets stronger and stronger and stronger as you go towards the range edge. If we look at MSAFLP, which is a pretty blunt instrument to look at DNA methylation, but good for a first look, we can see these regional differences. These are controlled tadpoles, and they're different. Their methylation status is different. Not surprising because they're genetically different. But if we look at within family, treated versus non-treated tadpoles, we also see differences in methylation. And so in these graphs, you can see the open symbols are the control tadpoles that were not exposed, and the filled, two different types of symbols in each graph were different families of suppressors, basically. So we had, our experimental framework had two different suppressor families. I'm showing you this because I want to make the point that the response to this is actually, it's complicated because it's both dependent on the receiver and the sender of that signal. So depending on who the suppressor is, you might get one response versus a different one. But it is changing methylation. We also looked at differential methylation in this experiment. So we used peak height data instead of presence-absence. And we only found one differentially methylated locus out of about 300. And that's what that locus looks like across populations. And you can see that it has affected. So the red bars are the treated individuals. So they have higher peak, so less methylation. But if you look at mass data, there's no relationship, right? So methylation's changing, mass is changing, but they don't really look like they're linked to each other. And we don't actually know causality here, right? We just know that methylation is changed when they're in this situation. We also know that predator responses elicit some really interesting things in, especially in amphibian larvae, right? So it's well characterized that they have this morphological plasticity. So these are not cane-toed tadpoles, sorry. These are not cane-toed tadpoles, but you can see that depending on whether or not they're in the presence of a predator, they change their morphology. We know there's also a behavioral plasticity in response to predator exposure in amphibians. And so we did similar experiments where we exposed cane-toed tadpoles to predator cues. And the way we did this is different than that conspecific exposure. We've actually taken a tadpole, put it in a syringe, and expressed it. And so the tadpoles, the chemicals in the cane-toed skin get emitted when it's damaged. So if it's being predated on, it emits these chemicals. And when you do this, you can simulate that predator experience for the tadpoles in which the tank that you're putting this into. And we then looked again at MSAFLPs in this context, and we saw similar things. So we do see changes in methylation in both of these different types of chemical exposure. And we started thinking, well, is this just a response to predation? Is this the same response? Like if you're exposed to an older conspecific, or if you're exposed to a broken tadpole skin, is this the same thing? Are you just responding to a predator? So keep that in your head for a second. I'll come back to it. So, maybe is the answer. DNA, does it mediate these phenotypic responses? Maybe. But we wanted to know if these responses would be transmitted across generations. And so this is where the Common Garden experiment came in that I mentioned earlier. So Rashmi ran this experiment as part of her PhD, and it was bigger than Ben-Hur. What she did was she used different methods to manipulate methylation in tadpoles. And the methods she used were that syringe thing that I explained to you. So that's the predator cue, but she's also used zebularine to demethylate. So it's a methylation inhibitor to affect methylation in these tadpoles. So we had these two different experimental approaches. And she took 24 family lines, 12 from the core, 12 from the edge. And she treated G1 offspring, and she had more than 4,000 animals in this experiment. It was crazy. And this is the way the experimental setup. So we had controls that had no exposure to anything. We had tadpoles with alarm cue exposure. We had those with zebularine exposure and those with both. And this was all replicated and enormous. And she raised them through the G2 generation. So the first thing we did, we were interested in what was happening with court values around metamorphosis. So we did an experiment to look at that. And what we found is that court was higher in range-edge tadpoles, so in controls, right, that were common garden raised. They come that way, right? They have higher court values. It was higher in tadpoles that were exposed to alarm cues and higher in tadpoles that were exposed to zebularine. We also used RRBS to look at methylation in these, and we found more differentially methylated regions on that range edge. In the population where we have less genetic variation, we found more of a response to these manipulations. We looked at the glucocorticoid receptor gene because we thought, oh, that's going to be a good candidate to look at. We saw no difference, no DMRs in that in the region of that gene. If you just have a look at these, you can see that this is the transcription start site. So exactly what you would expect, a drop in methylation. No response whatsoever to our treatments. We did look at all the other glucocorticoid receptor genes to see if we could find differentially methylated regions in those genes. And we found one. So SOX3 had a differentially methylated region at the transcription start site. And if you have a look at this graph, you can see that the treated individuals had less methylation than the controls. So what does this gene do? Well, we'll get to it. I also just wanted to share with you some other data from that experiment. So Rashmi did many, many assays on these animals. And one of the things she looked at was their feeding behavior and their mobility. And what she saw, probably unsurprisingly, if you read the amphibian literature, is that those exposed to alarm cues ate less and they moved more, moved less, they were more immobile. So they spent a lot of time just freezing, right? So what's going on here? Well, we tried our best to try to piece this together mechanistically, and here's what we think. So we know that predator presence decreases feeding. And we also know that it decreases anti-predator behaviors. And we know that that exposure increases court. So court itself affects feeding behavior. It decreases feeding behavior, just court exposure without alarm cues. That's not us, that's other work. And we also know that court itself will increase anti-predator behaviors. We also know that predator presence decreases SOX3 methylation. So that's what I just showed you. And we would assume then that that means that expression of SOX3 is increased, right? If it's in that transcription start site. We know that both court and SOX3 have a role in controlling obesity. And we know that SOX3 expression has this feedback loop with leptin. So they keep each other in check. And we know that leptin affects obesity, feeding behavior, and anti-predator behavior. That's as far as we've gotten down the mechanistic path for what's happening with this gene, but I think it's a reasonable argument that you might think this is actually real, right? Okay, so Rashmi also looked at mortality in those tadpoles, and she found some really interesting results. So these are data from the second generation, and what you'll see on these graphs are the treatment of the parents as well as the treatment of the second generation that are the focal individuals here. Not surprisingly, if you are not exposed to alarm cues, as compared to individuals that are exposed to alarm cues, there's a difference in mortality. So you're more likely to die if you're directly exposed to an alarm cue. However, if you compare individuals whose parents were exposed to alarm cues and they weren't, to individuals whose parents were exposed to alarm cues and they were, there's no difference. So your parents' experience of those alarm cues has some kind of protective effect that's passed on to their offspring so that they actually don't have this cost of mortality when they themselves and counter predators. We thought that was a bit cool. When we looked at zebularine, we saw the same thing, right? If your parents were exposed to zebularine and then you were exposed to alarm cues, no response. However, if you yourself were just exposed to alarm cues, you have this problem, right? So we also then looked at the impacts that our focal tadpoles, when they became suppressors to younger conspecifics, what did that exposure do to them? And what we found was this. So this is a little bit of a difficult graph, so I'll try to just walk you through it. This gray wedge shows you suppression ability, basically. So that's how badly you affect younger conspecifics, the wider that wedge is. And so what we found is that those individuals that were themselves exposed to alarm cues were better suppressors, right? So that their own developmental experience made them better at suppressing their competitors. However, we also found that there was a significant difference if their parents had been exposed to alarm cues. They were better suppressors, regardless of their own experience. So it does seem like something's being transmitted across generations. Now, obviously, we would have liked to have had another generation in this experiment so that we would know that we were having transgenerational inheritance instead of intergenerational inheritance, but honestly, it almost broke us to get this far down the line, so we don't have that. But I will say that one of the methylation manipulations, the zebularine, we did prior to the epigenetic reprogramming in cane toads. So we do think that it's actually like being transferred along generations. We also looked at behavior in these individuals, and we saw a very similar result. So here we go. We've got swimming behavior. If you were exposed to alarm cues, you swim more. And if your parents were exposed, there's no difference whether you were or were not exposed. So it really seems to be like pervasive, this effect. What about the methylation? Well, when we look at this experiment and look at the methylation, you see a big messy Venn diagram like this. I suspect that these are really, we would expect to see something crazy like this because we have so much genetic variation in the experiment, and we know that's driving a lot of the methylation status. But we do see this one gene, which in every case, range edge, range core, first generation, second generation, doesn't matter. That gene's always differentially methylated when those individuals are exposed to alarm cues. And so if we look at the DNA methylation levels of this gene, we see this effect where we have, these are the controls, so no exposure in either generation. These are the individuals who were exposed directly themselves, and their parents were exposed. And these are the ones, the red ones are the ones whose parents, I said that backwards. The black is only direct exposure. The red are the ones that parents were exposed, and then the offspring were or were not exposed. So we do see a difference in DNA methylation at this locus. These values are really low. And so, you know, when we look at this and thought, well, that's funny, we might have expected these to be in a different order, even though they're all less methylation. I think we're just at the bottom of the bucket here, so there's not really very far to go. There's hardly any methylation of this gene in those treatment groups. So this gene, we know it's important in sodium regulation. It's important for barrier function of the skin, involved in wound healing. Once again, these are things that we would completely expect to see affected if you were trying to control exposure to alarm cues, which come through the skin. Okay, so are these changes intergenerationally transmitted? Maybe sometimes they are. Okay, and finally, we wanted to know whether or not, that thing I asked you to remember earlier, whether the exposure to predator cues is the same thing as the exposure to older conspecifics. So we looked at the methylation data in that context, and we got this. And so the answer is probably not the same. There's very little in common, but I will show this, which is these two genes that I talked about earlier, in both the range expansion alarm cue exposure and range expansion suppression experiment. we see both of those genes differentially methylated. Why are they different? Well, there's probably a lot of good reasons why the results are not, why they are the way that they are. And so one is that the ecological context of these two cues is really different, right? One is, I'm the big older brother in the room and you better run. And the other is, help, there's somebody after me, right? So there's a different message being sent here. The developmental stage at which these things actually happen are different. So the conspecific suppression cue is only active earlier in the tadpole development than the alarm cue. So we're actually looking at different developmental stages here by maybe a week, which is about a quarter of the way through tadpole lifespan, and genetic effects, because these are separate experiments with different individuals with different genetic backgrounds. Okay, so the third thing that we think about is this idea about rapid adaptation to novel environments. We know that these novel environments, we expect they should have strong selection regimes, right? There should be all these new things happening that cause you to respond via selection. I'm not going to talk about like methods to measure selection, because all of you guys do this, right? What I wanted to talk about is experimental approaches to measuring this, and how we think about how to set up these experiments. Because we think about this a lot. So this is a paper that our collaborator, Natalie Hoffmeister over at Cornell, put together, where she looked at starlings. So we're talking about starlings now. So these are starlings that are native range ones from the UK, one's from the US, and one's from Australia. She's done these nice Manhattan plots. And you can see there are regions of the genome that look, when you compare native to introduced across both of those introductions, you see similar patterns in specific areas of the genome. Of course, there are areas of the genome where you see, in one comparison, you see signatures of selection, but not the other. There's lots of papers out there like this. This is a valid thing to do and useful. But, guys, we're excited because we're replicating invasions, right? But we've got a sample size of 2 still here. That's really what this is, right? And so what would be ideal is if we could end up with all of these populations looking at these kinds of comparisons. And I think that's the kind of thing that we all need to be working towards. The other approach you can take is one that Kat took in her PhD, where she's got native range samples and she's got Australian introduced samples. But we talked about the fact that, are we comparing the right thing here? Because actually things have happened to those native range samples since the animals were taken from there to bring to Australia. And so Kat went and got historical samples from the time period that starlings were taken from the UK and included those in her analysis. And so we could look then at changes in, selection regimes in the native range across time, but also across space. So this was a really nice way to look at this data, I think. And so Kat first started out with doing PCA. So here's the native range, and the historical are these gray dots. So you're like, oh, okay, they look pretty similar, not much has changed. Here's Australia, 2 really different groups of birds. You might be thinking, oh, selection in the introduced range. It's not. This is, yesterday, Rob was talking about things that look like selection that might not actually be selection. We found that a lot in the Starling introduction in Australia, and Kat spent a lot of time trying to pick apart demographic effects from selection. But we know that these are really just like founder effects from different introductions. So one in Adelaide and one around Sydney, different groups of birds that were brought. So when she looked at this a little further, she did some SNP analyses. And what she found is, if you have a look at the pink circles are UK versus the UK historical. So she found a similar, or if not more, SNPs under selection within the native range across time than she did between the native range historical versus Australian. So, this is like a warning that we can't just like take contemporary samples from the native range and think that we're comparing the right thing when we look at that between that and invasive populations. So, yay for historical samples. Let's do more of that, right? One of the other things Kat did was looking at morphology or like phenotype and trying to compare that to genetic background. So these are a couple of figures from a really complicated paper that you should read if you're interested in these things. And if you have questions, please ask Kat. But this is obviously a genome, and what she's done is she's taken different morphological measurements and done GWAS to pull out SNPs associated with those morphologies, then look for selection in those SNPs. And she's found some SNPs that are apparently under selection, and some of them are yay, even annotated, and put those on the genome there. In this paper, she also looked at variation. So this is really looking at mean values, but you can also look at variation around your phenotype, your genotype, and your environment. And that's actually something that you don't see very often, and it's really interesting what she's done here. So this is phenotypic dispersion. So she's collapsed her phenotypes down into one measure, and then dispersion is just that value that how different you are to all the others in your population, essentially is what that is. So it's a measure of variation, done it with genetic data, and then 2 environmental axes. And so these are also collapsed down from a bunch of bioclim variables. And this axis is primarily temperature with a little bit of rainfall in it. And this axis is primarily ground cover with a few rainfall variables in it. So when she looked at these data together, she found that, genetic variation drives phenotypic variation, no surprise there. But she did find this interaction, which was really interesting. So this is that relationship that you see where, the variation in your genetic background is driving that phenotypic variation that you see. That's this one. And that's when you have high variability in those environmental variables. So ground cover mostly is here. What's really interesting is that when you get low variation in the environment, that relationship between genetic and phenotypic variation completely breaks down. And so, we look at this as low plasticity and high plasticity responses, depending on the environment. Okay, so I did promise you that there would be a plot twist, and so we're going to get to that now. Our introduced environment's really novel. Well, we all like to think they are, but how do we know? What are we measuring? Are we accounting for everything? Definitely not. How do we know it's really novel? Cane toads are from Central and South America, as I said. This is Rhinella marina, so that's our species. It's the only cane toad found in French Guiana. It's the only Rhinella species found in French Guiana, to our knowledge, which is where we think they came from here. I'm telling you this because what I tell you next will make you wonder if we have multiple species. So, we all know it's important to go to the field and see our field site and understand it. And we definitely wanted to do this, but working in French Guiana is very hard. Getting permits and even travel, everything's hard there. Anyway, we eventually got the green light, we got the permits, and we went to look at cane toads in French Guiana, and we found this. So on the coast, there's these little cane toads that look very much like Australian cane toads. These are 2 adult pairs on the coast and in the rainforest, right? In amplexus, so they're adults. These rainforest ones are enormous, huge toads. And the sex ratio in the rainforest is crazy. It's like completely male skewed. And it's like 50-50 in those coastal toads. And we went, oh, what's that? That graph on the side there just shows you the difference in mass and snout to vent length, showing that they actually are different. Now, these toads are taken from areas that are not very far apart, 10, 20 kilometers, not far at all. If you look at movement data from the native range all the way across the invasion trajectory, you see this crazy thing where we've got, toads in Western Australia move the most, right? But in the native range, they don't move all that much. So, you know, they're in these areas pretty close to each other, and they're kind of just sitting there. They're not really mixing up very much. So what's the genetic story? What does it tell us is happening? Well, we've got 17 whole genomes that we've looked at so far, and we hope to get many more. But when we look at that, we see this horrifying result, which is that those introduced populations, which here are showing very low genetic diversity, are kind of genetically intermediate to the coastal and the rainforest toads. So what does that mean? We've always assumed that toads came from this lush jungle. They came to Queensland where everything's like wet and beautiful, and then they went across the mountain range and things got dry and they adapted. That's the story we've always like followed. But you know, and that's the data that shows. This is the precipitation data. We see two genetic groups in Australia. Queensland's very much like Hawaii. You get over the Great Dividing Range and all of a sudden we've got different looking toads, right? But maybe beaches in the native range are equivalent to linear deserts. Maybe those toads haven't adapted as much as we thought they had. And then, you know, this question, which is the horrific one, is have we created some super toad that has the ability to persist in both of those environments? And maybe what we see in Australia is some sort of sorting of those alleles that came here together with those toads. Probably the most parsimonious explanation, right? This is like making people think hard, right? This is a very big change in the way that we view this invasion. And clearly we need to better understand native range genetics. And we have lots of samples that we hope to be doing genome sequencing on soon to try to really pull apart what's going on here. So I'm just going to wrap up. I want to say that the things I think that we need to do going forward that are most important, I think it's this relationship between genetic and epigenetic variation. I think it's really interesting. I think it's got some legs, and I think we ought to be paying a little bit more attention to what's happening when you don't have genetic variation. You know, maybe this is a valid way to create variation. We need to go beyond these candidate gene lists. Like I do it, we all do it. It's a valid thing to do. We need databases with candidate genes in them so that we can learn and understand what's happening in genomes. But we really do need to do better. We need to do more manipulative experiments where we look to see what happens when we perturbate things like methylation or whatever. And we really need to strengthen our knowledge of those native range populations. So, you know, when you think about it, we all work on invasive species. Everyone's really interested in what's happening in the invasive range. Usually people are not at all interested in what's happening in the native range because it's a bit boring. We need that information. We really need to understand the population structure and the selection that's happening in the native range currently and historically, so that we can better piece together what's happening from an evolutionary perspective in the introduced ranges. So this is something that, you know, if we get $20 billion from Neil, I hope we spend some of it on this. Yeah, I'm done, and thanks for listening, and I'm happy to answer any questions that you guys might have about this.
Thank you, Lee. That was a fascinating talk. Do we have some questions from the audience? Just a reminder to do it in the microphone.
Thank you, Lee. That was great. So I'm curious about your epigenetic data, and in particular, I was just wondering if you'd passed out sex differences in epigenetic response.
No, the treatments were done to both males and females. Right. So yeah, I've written a grant to do that. Didn't get funded. We'll write that grant again, maybe. Okay. I think it would be worth doing. And
I missed what tissues you were looking at.
So there were whole tadpoles.
Oh, okay. Right. Yeah, again, if you could pass out tissue-specific responses, might be really interesting.
It might be. Cane toad tadpoles are very small, and we may not. Yeah, it'll be technologically interesting to try to do some of the stuff that we do with those tiny bits. But I think you're right. It would be nice if we could look at tissue-specific.
Yeah, super. Thank you.
Is there anything more known about the sex ratio bias in the rainforest, the basis of it?
No,
Nothing
it's,
at all.
wouldn't we like to go back there and look more? Yeah, I don't know what the answer is, but something's different about those toads for sure.
Cool, I can see we've got a few on Zoom, so we'll give them a chance. If you can pop up the window. Oh, okay, they're not questions. Questions in there? Yeah, Catherine.
So you preface my talk really well, so I'm very excited about that. Have you looked at getting methylation data from historical specimens?
Yeah, actually, Claire Holloway is, I don't know if you know Claire, but she's at CSIRO in Australia, and she's just developed some very cool technology to actually look at chromatin conformation in formalin-fixed samples. So not DNA methylation, but she's very keen to have a look at some of their historical cane toad samples and try to understand other epigenetic states.
I think that'd be really interesting historical data.
Look, I know that there's a guy I know in Adelaide that's been doing what you're talking about, and I've never tried to do it, but I reckon it's worth looking into. I think it would be quite interesting.
Formalin usually how your type of specimen is fixed?
In all honesty, cane toads are not well represented in museums because, especially in Australia, museums don't like invasive species. And historically, they didn't really keep them. I know it's so frustrating. It would be so nice to have those founders, right? But we don't. But I think the museum samples for toads that you'll find will be formalin fixed. But the starlings, however, are a different story, right? We've got toe pads we can work from.
Oh yeah, that'd be cool.
Yeah.
Yeah.
We need to go to Europe, and I understand there's more than 10,000 starling samples in museums in Europe, which would represent, a really nice spatial and temporal study that I'm dying to do.
Okay, I'm going to sneak in a question. Two questions. For the methylation work, one question is, have you thought about looking at telomere length? And the other is if you've thought about doing, of course you've done a lot and you've probably got a lot on your plate anyway, but have you thought about testing other types of potential trauma, like starvation or something like this, and the effects of that?
First answer is I've never touched telomeres. So, obviously read those papers, but I just have not gone there. Maybe in a world with more time, I would go there, but I think, I'd like to kind of nail this a little bit better first. Sorry, your second question was?
Any other types of potential trauma experiments?
Yeah, so the obvious thing is a nutritional manipulation, and some of our collaborators have done some nutritional things. We haven't actually done it in our lab, but one could do that pretty easily with tadpoles. I think the answer is we probably need to do some more common garden work, but I'm working up the nerves to actually do that, because that was a crazy amount of work and took 4 years.
If I may, I'm going to ask one more. So at the beginning of your talk, you said that you'd done the microbiome of the cane toads, and so in those two different size classes that you get in the tadpoles, do the microbiomes differ? And also coastal to rainforest, do the microbiomes differ?
Those are good questions, Neil. We've never done tadpole microbiome. We've only done adult microbiome. And no one collected guts from, so these are gut microbiomes I'm talking about. I know that Keith, what's his name? Keith Christian and Greg Brown have been working on skin microbiome in cane toads.
Right.
But no one collected gut samples in French Guiana, so we would need to go back to do that. It's interesting though. I think there's a story to look at the skin microbiome, gut microbiome data together, because when they shed, they eat their skins. And so we have this idea that maybe they're seeding their gut microbiome from the microbiome on their skin, but yeah, future.
Hi. With regards to the last question on other introduced strains, really novel, has there been any studies with regards to the environmental variables?
Yeah. So Dan Sletchnik, the first data I showed you, looking at population structure and genetic diversity across Australia, he did look at environmental variables to see how similar they were to the native range or not. But I guess my point is, you know, we look at things that are in BioClim because they're there and they're easy to download. But, you know, we don't know what is actually happening. It's really hard to like sort of capture all the selection regime that exists in a new place. It's hard to know what's really happening. You know, is it daylight? Is it? I mean, there's a million things it could be, right? So yeah, we've looked. I'm sure we've missed things.
And what was his findings in regards to that? Is that
Yeah,
different or similar?
so I should tell you this actually, because that work, when we did that work, we were thinking that the cane toads that came to Australia were coastal toads because, you know, humans are lazy and they probably just picked them up off the beach and threw them on the boat. And so that genetic diversity data that I showed you in the beginning, where we had lower at adaptive loci, we had lower genetic diversity in the native range. Those were only coastal toads. So, you know, now it would be worth going back and looking at rainforests and coastal toads to see what's happening there. So the environmental stuff that was done based on, you know, we've got this lush rainforest kind of area. We're coming to Australia. Queensland's quite similar, you know, and so it looks that way. The environmental data is similar in Hawaii, it's similar in Queensland. It changes significantly when you go west. But, you know, we were thinking lush rainforest, but we didn't realize we actually might have two separate environments in the native range that we need to think about.
How about in terms of morphology, the ones in Australia, do they still fall in between the rainforest and coastal ones?
Well, I mean, the morphology in Australia has been crazy because it, you know, I don't know if you've read any of the spatial sorting papers talking about like how dispersal speed has increased as they move westward. We know there's all these like morphological associations to that, like, you know, limb length and things like that. And so I think that it would be so confounded by that process that it would be really hard to like say much about how it fits into the two ecomorphs in the native range.